Well, thank you all for coming this morning. It's really hard to follow Kat. That was an amazing presentation, but it will be about fungi at least. And so I want to talk a little bit today about some of my group's work looking at manganese oxidation and the mineralization of manganese oxides by ascomycete fungi. And so manganese exists in 3 different oxidation states within environmental systems. So we have manganese 2, 3, and 4. And all 3 of these oxidation states have very important environmental and physiological functions. For instance, all 3 of these oxidation states have antioxidant activity, so they can degrade reactive oxygen species and decrease oxidative stress to organisms. The oxidized forms of manganese 3 and 4 are extremely strong oxidants. They're one of the few oxidants that can actually degrade lignin. And then also manganese is important for photosynthesis. It's a cofactor in photosystem 2, for instance. Manganese oxides exist in a wide range of different types of deposits within the environment. For instance, within the ocean, there are large areas, ocean basins, abyssal plain sediments, that are almost completely covered by manganese nodules. They can constitute about 90% of certain ocean basin floors. And these are little manganese, ferromanganese deposits that grow and nucleate over millions of years. You also see manganese crusts forming within the deep ocean. And within soils and sediments, we get enrichment of manganese within certain soil horizons, as well as the formation of small nodules within different soil aggregates. And many of you are probably familiar and have seen desert or rock varnish, which are manganese oxide veneers that form on surfaces. And you can get these in environments like this is a picture from Elkhorn Slough Estuary, where you get fluctuating water that can, in arid regions, that can form these little veneers on the surface of the sediment. You get rock varnish on rocks, and most recently, and excitingly, there's been manganese oxide veneers found on Mars. And so why do we care about manganese oxides themselves and the formation mechanisms? Well, manganese oxides have been coined the scavengers of the sea. They're extremely reactive compounds. So they have very reactive surface functional groups, high surface area, so they can absorb lots of contaminants such as lead, nutrients like phosphate. They're a reservoir for carbon. They can be used as a terminal electron acceptor for microorganisms that can metabolize organic carbon and couple it to the reduction of manganese oxides. And as I mentioned, they're also extremely strong oxidants, so they can oxidize recalcitrant carbon forms like humic acid and lignin. So despite the fact that manganese exists in a wide range of environments, and we know it's important environmentally and physiologically for organisms, we actually know very little about how manganese oxidizes within the environment. And so here I'm depicting some of the dominant manganese oxidation pathways that we know exist in this cartoon. And just to point out, unlike iron, when you have reduced iron like iron 2, you stick it in a beaker and put it in the presence of oxygen, it's going to precipitate spontaneously at circumneutral pH. So it's extremely labile. It's going to oxidize very rapidly. Manganese 2, however, is very stable. And you can have a manganese 2 solution sitting on your benchtop for decades, and it'll never oxidize. And that's because the manganese 2 ion is extremely stable. So you have to really distort the orbital symmetry of the ion for it to actually get oxidized. And so you need some sort of surface catalysis reaction. And a bunch of different compounds can induce this destabilization effects, such as organic ligands can complex to manganese, allowing for it to be oxidized by oxygen. Manganese can absorb onto mineral surfaces, and that will destabilize the manganese 2. As well as there's a wide range of microorganisms, fungi, and bacteria that can oxidize manganese 2. And so I should point out that we're taking manganese 2 to manganese 3. It's a 1 electron transfer step. Once you get to manganese 3, we don't really know what happens. Sometimes it's oxidized by these oxidants as well, or manganese 3 is very unstable when it's not complexed, and so it'll disproportionate to 2 and 4. Now recently, about 10 years ago or so, it was also discovered that there's another oxidant of manganese that doesn't have anything to do with direct oxidation by a molecular oxygen, but instead it's by the reactive oxygen species superoxide. Now superoxide is an oxygen radical that forms when you donate 1 electron to molecular oxygen, and it's extremely reactive with lifetimes of seconds within natural systems. So you can get superoxide formed in the photic zone of the ocean, for instance. When you have dissolved organic matter, it gets photoexcited, it donates an electron off to oxygen. You form superoxide, which rapidly oxidizes manganese 2 to manganese 3. This is an extremely fast reaction. So my group has been studying various aspects of manganese oxidation within different ecosystems as well as different organisms. And what I'm going to talk about today, however, is some of the work that we've been doing trying to understand manganese oxidation within passive limestone treatment systems for coal mine drainage. So we started this project about 12 years ago or so, where we were investigating the treatment of coal mine drainage in Appalachia. And so Appalachia right now has about 300 to 400 mines that are undergoing remediation. And this is because the coal, when it comes up, it oxidizes, it releases a lot of metals into solution. And the metals are primarily manganese, which can be up to molar level concentrations of manganese, as well as all the associated contaminants like lead and cadmium, arsenic and zinc, for instance. Now, it's been a very attractive strategy to try to use these kind of limestone beds to remediate the coal mine drainage. And these beds get laid down. The limestone brings the pH up to about 7. You throw in some carbon, straw, or compost, some nutrients for the organisms. You get them cranking. The microbes will oxidize the manganese 2 to the manganese oxides, which is that black color right here in the limestone treatment systems. And then because of that really strong adsorption capacity I talked about with the manganese oxides, all the metals will just stick onto the surfaces. And so you essentially clean the water. And so what comes out the other end is clean. Now what you have here is an ore body, so you can then mine that. You can pull up all the metals, you can extract them, lay down clean limestone, and start the process again. Well, like many things with bioremediation, some of these systems work, some of them don't. And we don't really understand enough about the systems to know why. So they're black boxes. We just throw down carbon, we throw down nutrients, we hope the microbes are there, and we let them go to town. But it's not working all the time. And so we started working on this to try to understand, can we actually deconstruct that black box and figure out what exactly is happening within these treatment systems to increase efficiency. So here are some of the questions that we've been addressing over the years. And I'm going to mainly just focus on the top one here today, which are who are the key microbial players within these treatment systems, and how are they oxidizing manganese? And then we've also been trying to address what are the optimal oxidizing conditions, what types of nutrients do we want to put down, what types of aeration, and so on. And then a big part of my group is really understanding the manganese oxide products. Are they reactive phases? Can we study their mineralogy in such a way that we can improve them to make them more reactive? But again, today we're going to focus on that top question. So what we found as we started studying these systems was that we had a really fungal-dominated ecosystem. Fungi are really running the show here, and this was a brand new thing for my group. At the time, we had most of us are geochemists, so even bacteria are a little terrifying. But now we had to deal with fungi, and what we found was we were getting these extremely fast-growing organisms that oxidize manganese incredibly fast. And they do it very efficiently and can withstand extremely high concentrations of manganese. So here are some of the faces of the manganese oxidizing fungi that we've isolated over the years. We have 12 unique isolates that we've obtained. 6 of them have their genomes now sequenced by JGI as part of our project. And I have those stars listed up here for a subset of those. And what you can see is that, well, first I should say that these are ascomycete fungi, so they're filamentous fungi. For those of you who aren't familiar with filamentous fungi, again, I'm a geochemist, so I talk about it in a very simplistic way, but I think of them as roots. So what we have is you essentially stab the culture in the middle here. They grow radially out like a root system. All the activities happening at the surface of the hyphae, and they oxidize manganese as they grow out. That brown to black color are the manganese oxides that are being formed. So you see there's a kind of a wide range of distribution patterns and growth patterns by these fungi. What we also found was there's quite a bit of diversity in the relationships between the manganese oxides that were being formed and the different cellular structures. So for instance, for many of the isolates, the manganese oxidation was happening right on the hyphal surface. So down here we have some SEM and TEM images where you can see these hyphae and hyphal cells. And these black manganese oxide deposits and these little round balls all over the surface, they're forming right on the hyphae surface. However, there was a lot of the other organisms that actually, the manganese oxides were forming at a distance away from the surface. And so they're forming away from the hyphae. So for instance, Pyrenicheta and Staganospora, which are going to be the two organisms I'm going to mostly talk about later in the talk. Here are the hyphae, and you see manganese oxides forming away from the hyphae, and they form these really beautiful, intricate manganese oxide aggregates that don't have any relationship with any cellular structure. And then lastly, one of my favorite organisms, this was Stobella, and it forms these beautiful reproductive structures where you can see the manganese oxidation is happening right at the base of the structure. Now, Stobella was the first one for us to start studying, one, because it was really pretty, and then two, that we know a lot about, well, we don't, but people have been studying fungal cell differentiation for a long time. And a lot is known about the mechanisms of how fungi differentiate their cells, how they form reproductive structures. And so in starting to dive through that literature, the one thing that really struck out at me and led us to start understanding what was going on with the manganese is the fact that when fungi differentiate their cells, when they branch their hyphae, when they form reproductive structures like this, they do an oxidative burst. So they form a lot of superoxide outside the cell, which, as we know, superoxide can be oxidized by manganese. And so this gave us kind of the first inkling of what potentially could be happening. So as a spectroscopist, the first thing we wanted to do was confirm where the manganese is within these fungal cultures. So the first thing I wanted to do was stick the Stobella under a beam. So we do a lot of work at synchrotrons, and so what we did was we said, okay, well, I want to see where manganese is distributed within those fungal structures. So we have a Stobella growing on the Petri dish. The beautiful thing here is we just stick it right in the beam line, and we can do X-ray fluorescence microscopy on that. And so by opening up the beam and looking at all of the energies, what we could do is map out where all the manganese is. And so here's that reproductive structure. See, manganese is concentrated right on the base, and there's a little bit up on the top where the conidia is. And then we could actually focus the energy of the X-ray, so we could specifically look at different oxidation states and get redox maps. So we could look at where all the manganese 2 is versus manganese 4, and then on this tricolored plot we have just potassium as our third end member. What you can see is all the oxidized manganese is on the bottom, but there is some manganese 2 being transported up into the top, up into the conidia. So what we wanted to do is say, okay, well, can we learn from what we know about fungal cell differentiation and the formation of reproductive structures to really confirm that maybe superoxide is the oxidant forming these manganese oxides. So in basing what our approach off of the literature, what we found was that there's a class of enzymes called NADPH oxidases, or NOx enzymes, that are involved in this oxidative burst that produce superoxide during cell differentiation. And so there is an inhibitor called DPI that you can use that will essentially shut down the activity of these enzymes and inhibit cell differentiation or the formation of these reproductive structures. When we did that, we get no conidia and we also get no manganese oxidation. So that gave us a hint that superoxide was essentially what's oxidizing manganese. Now then we could play some chemistry tricks on this to confirm this. So up here I have the chemical reaction for superoxide reacting with manganese. You form hydrogen peroxide manganese 3. Now we could just instead scavenge that superoxide. So we've done this with a wide variety of enzymes as well as metals. In this case, what I'm showing you is copper. So we can redirect electron flow. So what we want to do is say, okay, well, copper actually reacts much faster with superoxide than manganese does. So by adding copper to the system, I'll scavenge the superoxide away, and we should see a decrease in manganese oxidation, which is what we see. So we see as we add increasing amounts of copper, we shut down manganese oxidation, suggesting that we've redirected the superoxide reactant away from the manganese. And it wasn't a toxicity effect. We can add similar amounts of zinc, and we don't see that because zinc is not redox active. So for Stobella, what we're finding is that superoxide is this oxidant of manganese, and it's a function of the organism undergoing the formation of reproductive structures. Since that time, we've found that this is a widespread phenomenon with the fungi. For instance, we're coming back to Staganospora and Pyrenicheta here, where they're growing here on a plate, and in the presence of manganese, we can add DPI to inhibit the NADPH oxidase activity, and you see less manganese oxidation. Again, we can add copper, and again, we can scavenge it away. So what we're finding is this kind of universal process where what the hyphae are producing superoxide, that's oxidizing manganese, forming manganese oxides. And that's really that cell surface-associated manganese oxidation process. So superoxide's lifetime in these systems is microseconds. So it's not going to transfer away from the organism. It's going to be happening right at that surface. And so those manganese oxides on the surface of the hyphae can be associated with that process. But what about these manganese oxides that we're finding that are far away from any sort of reproductive structure? So for instance, here we have a hyphae, here we have a manganese oxide. There's no way the superoxide is going to travel that far. It's just too reactive. So really that's where I want to go for the rest of the talk, is to talk about that secondary pathway. So is there another mechanism that's responsible for manganese oxidation by these fungi? And we postulated that the organism was producing something within the secretome, it was secreting something out that was an oxidant of manganese and producing these manganese oxides. So where I'm going to go from here is summarizing very briefly a lot of the work that was done by Colleen Hansel, who's sitting in the audience, who was a PhD student in my lab and now a postdoc. I'm going to do only talk about a really subset of the data, but what Colleen did was she grew up 4 of our fungi that their genomes were sequenced by JGI. We harvested the secretome at 3 different time points in the presence and absence of manganese. We characterized the secretome oxidative capacity, as well as working with our colleagues at EMSL, did eye-track proteomics to be able to quantitatively determine the changes in the relative percentages of proteins over time and between the different fungi. And then we also used some gel-based techniques to try to start identifying what those manganese oxidizing proteins are, and I'll come back to that in just a little bit. So first of all, are these secretomes diverse that are being produced by the fungi? Are they different, and do they change over time? So here's just a summary of some of that eye-track data, and again, I'm not going to go into too much of the details, but just to say that we see a diverse suite of different protein classes that are produced by the fungi. And if you look at this broad class level, you don't really see any big changes either over time or between the 2 different fungi that we're going to focus on for this talk. However, if you look at the changes in specific proteins within the secretome over time, so here what you're looking at is the change or the increase in different proteins between 7 and 14 days and between 14 and 21 days. You can see that the proteins are changing. The relative percentages of the proteins are changing over time, and those distribution patterns are actually different between the 2 different fungi. So these organisms are obviously regulating their secretome differently. So we were thinking, well, this is going to tell us something about how the secretome activity is going to be changing. And Colleen has spent a lot of time characterizing those different proteins. And again, I'm not going to talk about this on a broader scale today, but if you'd like to talk with her, I'm sure she'd love to talk about it. But mostly what she's finding is that there's a big changes in the carbon degradation enzymes over time, as well as just some fundamental differences in the physiology of the different fungi. But to bring it back to manganese, what we did see is that we also do see differences in the secretome activity between the Staganospora and the Pyrenocheta. So Staganospora, for instance, when we harvest the secretome at 7 days, it's got a very strong oxidative capacity for manganese. It oxidizes a lot of manganese. 14 days less so, and less so at 21 days. So that oxidative capacity declines over time. Whereas Pyrenocheta, we really didn't see the secretome oxidizing manganese at all at 7 days, and most of the time we didn't see it at 14 days either. Sometimes that varied a little bit. But then at 21 days, the secretome starts to become active. So the next question was, well, is that an enzyme in there that's just changing over time? So the first thing was to confirm that manganese oxidation was linked to enzymes. So what Colleen did was she took the secretome, she boiled it, added a protease, and confirmed that yes, if you shut down enzymatic activity, that you're not getting manganese oxidation. So it's an enzyme that's oxidizing manganese. So for the first question that we wanted to ask was, well, is this linked back to superoxide again? So we have shown previously before in a bunch of the bacterial systems that we're studying that bacteria produce extracellular enzymes that make superoxide that oxidize manganese. And that's a dominant pathway that we're seeing for bacterial manganese oxidation. We wanted to know if the same thing was going on again. So is this system all driven by superoxide, which would have been wonderful, but that wasn't the case, unfortunately. So instead, when Colleen added superoxide dismutase to these secretomes, superoxide dismutase selectively scavenges superoxide. What she saw was no change in the oxidative capacity of that secretome. So manganese, the enzyme that's oxidizing manganese within these secretomes by the fungi is not linked to superoxide. So it's a fundamentally different pathway than what we're seeing right at that hyphal surface. Now that's not to say that there aren't other reactive oxygen species involved in this manganese oxidation. For instance, hydroxyl radical is known to be formed by a lot of the enzymes that we're seeing within the secretome, and hydroxyl can also oxidize manganese. And this is something that we're starting to dive into right now, is trying to see what are the other oxidative radicals that could be involved. But we can say that it's definitely not superoxide at this point. But we wanted to see, well, can we start figuring out what enzymes are there? And so this is a NADPH gel approach that's been widely used for studying manganese oxidizing organisms, where you can extract the proteins, in this case, all the enzymes within the secretome, run out through a gel. The enzymes are not denatured, so they're still active, and then size fractionate them out. These two lanes here, it's stained with Kumasi, so you can see just the total proteins within the secretome. And then we add a colorimetric reagent called leucoberberlin blue. Leucoberberlin blue only reacts with manganese 3 and manganese 4, so the oxidized forms of manganese. And when it does, it precipitates and forms a blue precipitate, ironically the same color as the Kumasi. And then, so now we know where those manganese oxidizing enzymes are, so we can cut those out, clean them up a little bit, and then we sent them off to our colleagues at EMSL for LC-MS sequencing. Okay, so to come to the results, what we found is a diverse suite of enzymes that are being used by these organisms that vary both over time for Staganospora and between the different enzymes. So unfortunately, we didn't have one single enzyme that popped up. Instead, we have a few. So we're still trying to figure out which or all of these are responsible for that manganese oxidation. Now, I should note that manganese oxidation has been studied for a very long time in basidiomycete fungi. Most of basidiomycetes take manganese 2 to manganese 3 and stop there. And the manganese 3 gets complexed to oxalate or citrate, and then that's a very strong oxidant for degrading lignin. So for instance, this is widely studied in wood rot fungi. Ascomycetes, however, and lacases have also been found in basidiomycetes. For ascomycetes, only lacase-like multicopper oxidases have been shown to oxidize manganese. And many times it goes manganese 2 to manganese 3, but sometimes it can also take it all the way to manganese oxides. So these multicopper type oxidases, they're similar to this bilirubin oxidase that we're finding here, so that wasn't a big surprise to us that we would come up with a bilirubin oxidase. But however, we find this tyrosinase and a glycal oxidase, as well as these FAD binding oxidoreductases that were pretty unique here in the system and that we're excited about studying. Because this could be expanding the different types of enzymes that are involved in manganese oxidation within fungi, within the ascomycete fungi. So this is kind of where we're at right now, and Carolyn's now trying to figure out how do we target which of these enzymes is responsible for the manganese oxidation within those gel bands. Can we narrow it down? Is it all of them? Are they acting in cooperation, or are they each oxidizing manganese? So this is data that's kind of hot off the press that I think Carolyn did last week, so I probably won't describe it properly. But what she's trying to do is start to add different substrates that will specifically target these different enzymes within those secretomes so that we can try to see how that's going to impact manganese oxidation. And so on the left here, we have Staganospora, and the enzyme targets, again, we're looking at this bilirubin oxidase and a tyrosinase that we're seeing in the gel bands. On the white, we have this is total manganese oxidation, or the percentage. And what you can see when you add bilirubin, you see a decline in the amount of manganese oxidation that happened within the secretome. And I should point out that the concentration of bilirubin ended up being a lot less than we had initially wanted because it was difficult to dissolve. So those concentrations are actually quite low. So we need to repeat that at higher concentrations. So the fact that it didn't completely decrease the manganese oxidation could be a function of that, or it could just be that, that there's something else at play. We added L-tyrosine, and you see that there's, again, a decrease in the manganese oxidation, but not completely. And then we added L-dopa, which we believe is a target for both of these, completely shut down manganese oxidation. So we're starting to see that we can start trying to identify what the substrates are for the enzymes, try to start targeting that. Carolyn has a whole suite of other substrates that she wants to start targeting and trying with these different secretomes to see whether or not we can hone in on that a little bit better. But then we look at pyranakita. Here our enzyme targets are these oxidoreductases, these FAD binding proteins. We added bilirubin, we shouldn't see any difference here because the bilirubin oxidase wasn't identified, and we don't see any difference if you take into account the error here. But we added DPI, which again is that broad spectrum oxidoreductase inhibitor. And what we do is we see a large decrease in the manganese oxidation, which would be consistent with these enzymes. Now moving forward, again, Carolyn's going to continue to try to start looking at the different substrates, start narrowing this down, as well as we're going to try to start size fractionating out these proteins. Can we get them into smaller, can we separate the proteins out, ideally purify them, and then be able to see which ones are contributing to manganese oxidation, if all of them are contributing, and to what degree. So to kind of wrap it up, what I showed you is that we have a large diversity of manganese oxidizing ascomycetes that are present within these limestone treatment systems that are contributing to the manganese oxidation. We now know that we have to target these specific enzymes for trying to increase the efficiency of coal mine drainage remediation. And you're going to treat these organisms much differently than if it was a bacterial-driven system. So this is giving us an idea of how to move forward. We've identified a large suite of new manganese oxidizers. We've also shown that we're starting to identify what these manganese oxidation pathways are, including this, a number of different oxidants, so NADPH oxidase, or this NOx-derived superoxide that's forming right at the hyphae. Interestingly, we're finding that in a lot of bacterial systems, a good majority of manganese oxidation can also be linked back to superoxide production. And so we're seeing kind of the synergy between fungal and bacterial oxidative pathways, which is kind of exciting to further investigate. But we're also seeing that there's these enzymes that are being secreted within the secretome by the fungi that are oxidizing manganese that has nothing to do with superoxide, may have other ROS, but we believe that at least we have some direct enzymatic manganese oxidation. Otherwise, we wouldn't have seen it happening within those gel bands where we had no substrates added. So the characterization of these reaction mechanisms is ongoing, and hopefully we'll continue to hone in on what's actually happening. But to kind of step back, we're really interested in what is the role of this process for fungi. So in basidiomycetes, it's very clear that the manganese peroxidase is acting on manganese, forming manganese 3 to degrade lignin, and that's very, very well studied. But ascomycetes, we don't know if this is the case. These fungi are growing on acetate, and yet they're making a lot of manganese oxides, and they're oxidizing manganese. So is there another function for manganese oxides, or manganese oxidizing organisms? And in general, we know very little about why bacteria also oxidize manganese. It's not used for energy, and so trying to understand what is the physiological point of oxidizing manganese and making enzymes to oxidize manganese if there's no real benefit to the organism. So we're really trying to start to target that in both bacteria and ascomycetes. And I know I'm out of time, so I'll just wrap up by acknowledging the people in my lab who have worked on fungal biomineralization and manganese oxidation. Most of the work I showed was from Carolyn Zeiner, and again, please go seek her out and talk with her. And Cara Santelli, who started this project with me when she was a postdoc in my lab. And then I want to thank JGI and EMSL, and Susanna mentioned that I was one of the FICUS awards, and this has been really instrumental for being able to do this work, being able to sequence these genomes, doing all this proteomic analysis has really taken this project much farther than we ever thought it would go. And with that, hopefully I'll have some time for some questions. Thank you.
Yeah, great talk. Thank you. So back to Stabella, beautiful image.
Yeah.
So these dark spots at the base with this high concentration of manganese 4.
Yes.
Do you think this dark color is a result of oxidation, or there is also some processes involved in secondary metabolite production, like melanin, linked to these oxidation processes?
So in the absence of manganese, we don't see that dark coloration, which is why we did the spectroscopy work to confirm that was manganese 4 at the base. So unless they're forming the melanin in response to the manganese, so then it would be hard to kind of tease that out. We think it's primarily just the manganese at the base. Yep.
I have a follow-up question. In Stillbella, when you quench the oxidative burst, do you lose production of those reproductive structures, the cinemata?
Do we lose the reproductive structures?
structures?
Yes, exactly. So that image I showed, you could see all the hyphae, but you couldn't see any of the reproductive structure. Sorry, I didn't point that out. Yeah.
All right, anything else? Oh, we've got one more over here.
I don't know which way to look.
So, hi. Is the production of these manganese oxides during spore formation, is it a cause or an effect? And I also know those spores are formed in a circadian fashion. I used to work on Neurospora, so I'm just curious about that.
Yeah, those beautiful Stillbella rings, at first I thought that was a circadian rhythm-driven process, and it turns out it wasn't. It was just, it was a surprise to me too. I wrote my entire career grant on that, and it turned out to be wrong. What was the first part of the question? I'm sorry.
Is it a cause or an effect
of
the sporulation?
The manganese oxidation. So people who study manganese get really mad when I say this, but I think manganese just gets in the way. So I think what the organism is trying to perform the reproductive structure, it's doing an oxidative burst, and manganese is really reactive with superoxide, and it just gets in the way. And so it oxidizes with the superoxide. And what we actually see is if you keep increasing the manganese concentration, you can shut down the formation of those reproductive structures as well, because we've scavenged all the superoxide. So the reaction rate between manganese and superoxide must be faster than its ability to be a signaling molecule for the conidiaphore formation. Yeah.
All right, I want to make this the last question over here.
So I may have missed it, but why do you get the oxidation at a distance in that second pattern you showed? It seems like you'd get a gradient that would be strongest right near the hyphae because that's where it's being secreted. So why is it nucleated further away?
Yeah, so that's another great question. So I think it comes down to the fact that you're forming manganese 3. So I think what happens is the secretome, or what's being secreted out of the organism, is forming manganese 3. And it's happening far enough away that it can transfer just a little bit far before it nucleates and then precipitates out. Whereas we don't see that right on the surface of the hyphae. We see that precipitation right on the surface. So I think there is probably some gradient. We just haven't studied it in enough kind of a spatial dimension to be able to see whether or not you would see this kind of gradient of oxidation away from the hyphae. Does that answer your question? Or no? Okay.
All right, well, we have a break now. We're a little behind schedule, but let's try to meet right back here at 10:30 and get started with the next few talks. And let's give a hand for all the morning speakers.